Hi, my name is Jeremy Schmutz, and I'm a faculty investigator at the Hudson Alpha Institute of Biotechnology. We work as partners with the DOE Joint Genome Institute on DOE mission-relevant projects, including plants and fungi related to biofuels and carbon sequestration. Why did we sequence soybean? Soybean is the major source of biodiesel in the U.S. today, and is grown in many other parts of the world also for oil production. It's also used as a fuel in many industrial processes and in food preparation. From the genome sequence, what we hope to be able to get is a reference sequence that's available for researchers to use. They can organize their own genomics research around for two particular uses. One is to increase the yield of soybean overall. In the past, soybean yields have increased primarily through selective breeding, sometimes called marker-assisted breeding from the genetic side, but even just the farmer picking out two different soybean plants in the field that have good yields and breeding them together in the next generation to produce a new plant. Now, what's happened is, in the recent years, we've kind of tapped out on traditional breeding in soybeans, and we can't seem to increase the yield anymore. And so a lot of the focus has now shifted to using genetics, and in this case genomics, to build a reference genome for soybean, and then be able to go in and actually breed specific genes, identify the causes of phenotypical traits in soybean that help with yield, like, for example, drought tolerance, or insect resistance, or pathogen resistance, or even just, more seeds or larger seeds, and then be able to breed those back into the soybean lines that the farmers plant here in the U.S. The Soybean Genome Sequencing Project is the largest plant project done to date at the Joint Genome Institute. It also happens to be the largest whole genome shotgun plant that's ever been sequenced. So we sequenced the soybean genome using a whole genome shotgun sequencing approach, where we took the approximately 1.2 gigabase genome and broke it up into lots of very small pieces, and then computationally reassembled the genome sequence like a big puzzle to put together a complete picture of what the genome of soybean actually looks like. And one of the interesting pieces about soybean here is that it actually is not just one genome, because it had in its past a whole genome duplication where complete entire copies of the chromosomes were all made. So soybean went from 20 chromosomes to 40 chromosomes, and then over time it's gone back down to 20 chromosomes again. What we found with soybean was that the retained difference between the two copies of soybean is very high, in that most of the soybean genome occurs in 90 to 95% identity in two occurrences in soybean. And on the gene level, there's still 75% of the genes. That means two copies of every gene are retained from the last duplication, which was approximately 13 million years ago. This is actually very important because in using soybean in the soybean genome here as a tool for crop improvement, we have to be very concerned about having two copies of the same gene extant in the genome, perhaps having the same function, although we haven't looked in detail enough to know what the differentiation of function is in all of the duplicated regions in soybean. From the community's perspective, one of the most useful things coming out of the soybean project is that the genome sequence forms a unifying reference where, just like if you had a map of North America or a GPS system and you could tell people exactly how to get somewhere, you can also tell people exactly how to get somewhere on the soybean genome now that you have a reference. And we can present the reference sequence and start layering information on top of it. So two groups working on similar kinds of pathogen resistance, for example, in soybean, now can tell each other how they're progressing and where they're trying to get to. So we can eliminate a lot of the duplication that's been going on from basically everyone working in a big black box of what the genome looks like. Now we've actually got specific base pairs to attach and hang off these disease-resistant genes and the yield traits in soybean.